ಬಟ್ ಇಲ್ಲೇನು ಯೂಟ್ಯೂಬ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಬೇಕೇನು? Hello everyone, myself is Dr. Avinash Amangi. I am from Gurudev Academy, Dharwad. So today we are going to solve the KCET question paper 2025, which is related with what biology subject, right? So as the first question, if you see, the cell wall is less prokaryote among the following is, right? So they have given option, that is cyanobacteria, mycoplasma, bacteria, blue-green algae, right? So here if you find out, the correct answer is what? The mycoplasma, because mycoplasma, even we call it as the smallest cell overallly, and which doesn't have what? The particularly cell wall, right? So it has only the particularly cell membrane, which is actually made up of something called as what? The particularly lipoprotein, right? So composition based, right? Then second question is the graph showing the concept of activation energy of the particularly enzyme is given below, right? So with respect to that, if you find out majorly like related to this particularly the activation state of what? Energy, and even with respect to that particularly enzymes. So we have to find out actually what here? The M and N, right? M as well as what? N. What does this particularly? Observe the graph and choose the what? Correct option for the what? M and N. So if you find out overall with respect to the enzyme and the substrate, as enzymes are specific to the substrate and they are going to accelerate the rate of what? Reactions. So when there is no any enzyme, right? So definitely substrate will reach to the what? The transition state, right? Then it will get converted into form of particularly product. So it required what? Lots of time. So with respect to the enzyme, if there is a presence of an enzyme, then the substrate and enzyme start getting react, right? So it won't get up with respect to the what? Transition state, and that will lead into the what? Formation of what? This particularly short graphical representation. And what does it means? The enzyme started reacting with the substrate and started converting into the what? Product. So based on that, M and N if you consider, that is nothing but what here? The M is activation energy without enzyme, right? So only it reached what? Maximum transition state. And then the N is the activation energy with enzyme. So it reached what? The less transition state and formed a particularly product. So here answer is what? 3, right? Then next question, that is what? Third question. Match the stage of what? Prophase 1 given in column 1, right, with their features in the column 2, and choose the correct options from the what? Choices given below, right? So we have to find out first what? Prophase stages they have mentioned. With respect to that, whatever the changes does occurs with respect to this, that what we have to find out. So here if you consider majorly the liptotin, what we are going to see here? Majorly here the chromosomes visible under light microscope, right? Then if I consider zygotin, there we are going to find out what? Chromosome starts pairing together, where the majorly synaptonemal complex formation start taking place. Then patchatin if you consider, with respect to the patchatin, that is nothing but exchange of the genetic material between the non-sister chromatid of the homologous chromosome, right? Then with respect to the diplotin if you consider, diplotin, what we are going to find out? Dissolution of what? Synaptal complex will begin, right? So that is what you are going to find out with respect to the diplotin. So overlapping dissolution will start with respect to the what? Diplotin. And then the the dikinesis if you consider, that is nothing but what? Terminalization of what? Chiasmata is going to what we are going to see it into the dikinesis, right? So majorly here if you consider, the answer is here the 2, right? So based on what? Comparison and then the what? Matching with respect to the what? Column 2. So answer is what? 2, right? So you people can compare with respect to the what? The A, B, C, right? Based on what? Seeing this particularly type of what? Video, right? So video based, find out the question and then find out the what? Options regarding with respect to the whatever the A, B, C type of what? The question what we what you have got, right? Then next statement is what? Question number 4, that is read the given statement and choose the correct option, right? So here in Kelvin cycle, first statement what they say is, in Kelvin cycle carboxylation is catalyzed by PEP carboxylase. Statement 2 says Hatch and Slack pathway, that is carboxylation is catalyzed by RUBP carboxylase, right? So with respect to that, if you consider overallly, right? So majorly if you find out, right? So in Kelvin cycle, if you consider with respect to the Kelvin cycle, right? So first carbon fixation, the atmospheric carbon fixation is carried by means of what? The Ribisco enzyme, right? So Ribisco enzyme itself is responsible for the carrying out what? The carboxylation. So with respect to the statement in Kelvin cycle, if you consider what they have mentioned, the PEP carboxylase, right? So RUBP carboxylase or Ribisco enzyme, that itself is going to catalyze and forms a first primary product, that is what? 3-peg, right? So or what we call it as phosphoglyceric acid, right? So that it's a 3-PGA, right? So 3-PGA, right? 3 molecules of what? The 3-PGA, that is phosphoglyceric acid, is formed after the first carbon fixation, right? So there the actually who comes? Ribisco, but here they have mentioned what? The PEP carboxylase. Then Hatch and Slack pathway, carboxylation is catalyzed by RUBP carboxylase, what they are saying. But here actually who is going to come? The particularly what? The PEP carboxylase, right? Here majorly who is going to come here? The PEP carboxylase enzyme, right? So this PEP carboxylase forms a primary product, that is nothing but what? Majorly the oxaloacetic acid, which is nothing but a 4-carbon compound, and this is nothing but what? 3-carbon compound. So even a Kelvin cycle can be considered as what? C3 cycle. Hatch and Slack pathway can be considered as what? C4 cycle. So with respect to that, here both the statement 1 and statement 2 are false, right? So that is what option 2 here, right? Then a TCA cycle, next question is what here? The particularly TCA cycle, right? So related with the TCA cycle, whatever the acetyl CoA, right, majorly like what? Starts with the condensation of what? Acetyl group with, right? So majorly what they are saying here, the TCA cycle starts with the condensation of what? Acetyl group, right? So what I know is the majorly the acetyl group is actually formed, or acetyl CoA enzyme formed with respect to the cytoplasm, because pyruvate cannot enter inside, directly cannot enter inside the particularly mitochondria. To enter into mitochondria, it should get converted into what? This particularly acetyl CoA, and that itself what I know is acetyl group, condensation of what? Acetyl group. With whom it is going to take place? It is majorly going to take place oxaloacetic acid, right? With respect to the what? Oxaloacetic acid. Then it will convert into what? The citric acid, right? So that is nothing but what? Mainly tricarboxylic acid, or what we call it as the citric acid, right? Which forms majorly here enzyme comes is which one? The citrate synthase, right? So citrate synthase itself is going to lead into the what? The formation of citric acid. How? By means of utilization of what? Acetyl CoA, that is what we call it as condensation of acetyl group with whom? The particularly oxaloacetic acid, to form what? Citric acid. So here answer is what? Oxaloacetic acid, right? Then next question is that sixth question, that is match the plant growth hormone with the column 1 with the suitable chemical derivatives present in the column 2, right? And choose the word correct option given below, right? So we have to find out from where actually these particularly hormones are produced, or these hormones are the derivative of what, right? So majorly you consider here abscisic acid. Abscisic acid is the derivative of what? The carotenoid, right? Abscisic acid is the derivative of what? Carotenoid derivative. Then gibberellins, if you consider, it's a derivative of what? Terpenes, right? Then kinetine is a derivative of what? Here the particularly adenine derivative, right? Then auxin is nothing but what? It's a derivative of indole acetic acid, right? So based on this particularly match, if you consider, the option E, first one is correct, right? Then the particularly next what they see is a respiratory mechanism controlled by medulla oblongata can be altered by. Now who can alter? If you consider pneumotaxi, that is the particularly a part of what the pons, right? So with respect to that only what it can do, it can control the particularly respiratory rhythm. Then chemosensitive area of what pons, if you consider, right, which is a part of what medulla oblongata, understanding, which is a part of what medulla oblongata. And what it can do, it can carry out the particularly control of what pH as well as what CO2, so that what proper rhythmic regulations can be what taken with respect to the what respiratory. So here if you consider, the option 1 is what correct, right? Then which among the three layer of what blood vessel, that is 8th question, which among the three layer of what blood vessel wall, tunica intima and tunica media and tunica externa is comparatively thin in veins, right? They are only asking related with respect to the veins. If you consider related with whatever these three layers, majorly if you consider tunica intima and tunica media, these both of them are what thin layer, right? But comparatively with respect to the what vein. So when I compare with respect to the what arteries, then definitely these are the what thin layers, right? But they are asking with respect to the what thin in the particularly what with respect to the what veins, right? So if you consider, the thinnest one is nothing but what, it's a tunica media, right? So that is what related with respect to this particularly 8th question, right? Then we'll go ahead with what, a 9th question, right? What they are saying here, the nephron transports a substance like what sodium chloride and urea is facilitated by the special arrangement called as what counter current mechanism, right? And that compares. So what we know is with respect to the word glomerular, right, glomerular filter, then what we are going to find out the particularly PCT, and with respect to that word Henle's loop, right? Then we'll have whatever this particularly DCT, right? So then lastly we'll have something called as what the collecting duct, right? So like that of what structure you are going to find out nephron. So mainly this particularly Henle's loop is this Henle's loop, right? Henle's loop is surrounded by means of what majorly like which will help in particularly counter current mechanism, and it is with respect to the what vasa recta, right? So that what the counter current mechanism can be brought, where the majorly reabsorption of what molecule can takes place, which are actually useful for the remaining process or filtration of this particularly urine or the blood you are going to find out, right? So next question here is, that is with respect to the 9th question, if you consider answer is what here 3, right? Henle's loop and the particularly vasa recta, right? Then 10th question, if you consider, that is the mechanism of muscle contraction or shortening of muscle, the dash get reduced and whereas the dash retains with respect to the what, the particularly length, right? So here if you consider majorly what we know, there is what A band, then there is a Z band and there is a particularly I band, right? So if I consider A band, which is actually made up of what, the myosin filament, right? And Z and I is made up of what, actin filament, right? So with respect to this only, if you consider, this A will not change its something called as what shape, it written as it is, right? But Z and I if you consider, right? So answer wise if you consider, in the mechanism of muscle contraction or shortening of muscle, the dash I band get reduces whereas the A band retains the particularly what length. So this is going to retain its length, this is going to change its length, and with respect to the what Z if you consider, majorly the sarcomere basis, it can become what shortened but it won't change its length. So that what you are going to find out. So majorly like what, who will retain, right? So who will retain the length, that is nothing but what, the particularly A band, and the who is going to get shortened, that is I band. So here answer is what, the particularly 10, right? Then identify the particularly correct sequence of action potential as it arrives at the axon terminal from the what choice given below, right? So if you find out with respect to the axon, what we are going to find out, the first one is what, axon terminal, followed by means of synaptic vesicles, then post synaptic membrane, then the particularly synaptic cleft and post synaptic neuron. So here that is what 11th answer is what the 2, right? So majorly like next question is what the 12th one, that is identify the statements given below that does not corresponding to the what functions of cortisols, right? Majorly here you consider which maintains the cardiovascular system of the kidney functions, right? Then if you consider it won't maintain the particularly what the which does not corresponding to the word cortisol. So majorly what cortisols can do, it maintain the cardiovascular system and the kidney function, so which is actually correct. Then produces what anti inflammatory reactions, yes, this is also correct, right? Then maintains electrolyte balance, osmosis and blood pressure. So here this one is what wrong actually, right? Then suppresses the immune response, yes, this is correct. Then stimulates RBC productions, right? So this is what actually wrong here. So with respect to this particularly question, answer is what the particularly 4, that is 3 and 5 is actually wrong, which does not corresponding to the what functions of corticosols. Then next question what they are saying is 13th one, when the pollen grains of flower of a plant pollinate the stigma of what flower of what another plant, so it is called as what? It is majorly known to be what? Xenogamy, right? So zeotenogamy, if you consider the transfer of pollen grains inside with respect to the what same plant of a different flower, right? So with respect to the autogamy, if you consider majorly what we are going to find out here, the transfer of pollen grains with respect to the what same kind of a what flower with respect to the same plant only. So based on the particularly question, when the pollen grains of a flower of a plant pollinate the particularly stigma of a flower of another plant, they are saying another plant, so definitely it is nothing but what? The xenogamy, right? So next question, that is related with what? The particularly 14th questions mainly, which is related with respect to the what? Double fertilization, right? So fusion of what? A male gamete with a central cell, right, in the what embryo sac of an angiosperm is called as what? The particularly is called as syngamy, apomixis, double fertilization and triple fusion. So when I see the structure of what? The particularly embryo sac, with respect to that what I find out at the microphylar end, I'm going to get the particularly 2 synergids, 1 female gamete, right? Then at the chalazal end I am finding out the particularly antipodal cells, right? At the center there are what? The particularly 2 polar nuclei, right? So firstly what is going to happen, one of the male gamete comes and get fused with what? This particularly female gamete, and majorly what I am going to call it as syngamy, right? But they are asking what here? The fusion of what? Male gamete with the central cell in the embryo sac. Now with respect to the what? This is a main cell, this is what with respect to the central cell. Now what is going to happen? One of the male gamete will get come and get fused with respect to this, and this phenomenon we are going to call it as triple fusion. What we call it as the triple fusion. So with respect to the question, the answer is what here? The triple fusion, right? Then here they have shown majorly the structure of what? The particularly pollen grain. So based on that I have to what? The particularly answer, right? So which of these option is true in the context of what? Above diagram of what? Pollen grain. So structure of what? Pollen grain, what we know is, if you consider the outermost layer is known as what? Exine here, right? And the innermost layer what we are going to call it as intine here, right? And then cell wise, if I find out this A cell, a large with what? The particularly irregular kind of what? Nucleus, and which has which can act as what? Reserve food material, right? And that is what? Large cell, which we are going to call it as what? The vegetative cell here, right? It is known to be the what? Vegetative cell, right? With respect to the A, if you consider, it is a vegetative cell. Then B, which is a small cell having what? Majorly the male gametes, and that we call it as generative cell, that we call it as generative cell, right? So with respect to that itself, if you consider, answer here is what? A is a vegetative cell with abundant food reserve, right? So which is larger in size with respect to that what? Irregular kind of what? Large nucleus and abundant reserve food material, what it can be like. It should provide a nutrition to the what? The generative cells which carries actually the male gamete. So B is what here, generative cell which forms what, the male gamete. So here answer is what, 2, right? Then next question, that is what, again match the following, right? So the hormone with its what site of what particularly production, right? So here mainly HCG and HPL, right? So mainly what we call it as the human chorionic gonadotropin, and HPL is the human placental lactogen hormone, right? So which is actually, name itself will tell you, placental. So here you can find out A is nothing but what, it is related with the placenta. Then progesterone which is related with respect to the what here, the corpus luteum, right? So formation of corpus luteum, then the particularly what is actually initiated with respect to the what, progesterone, right? Then you consider majorly androgen is related with what, Leydig cells. So Leydig cells which are present with respect to the what, testis. So there the androgens comes. Then relaxin, if you consider relaxin, that is nothing but what, known to be here the ovary hormone, right? Which is actually not mentioned much, but you consider relaxin, it actually help with respect to the what, overall development with what, even ovary too, right? So that what, with respect to the 16th question, answer is what here, the particularly 3, right? So that is what the match the following, right? So next question, that is nothing but 17th. So choose the particularly correct sequence of what, sperm transport during the ejaculation, right? So the releasing of the sperm, right? That what we call it as ejaculation, right? So with respect to that, how actually it is going to move with respect to the what, the male reproductive structure. So male reproductive structure has what, seminiferous tubules, right? With respect to the seminiferous tubules, what we are finding out, the rated testae, and which have what, the epididymics, right? So 200 seminiferous tubules will find out, then the rated testae, which from where the actually passes, there the actually sperm is going to move, which will reach to the epidemics to it will reach to the what, vasa efferentia, then comes to the what, vasa deferens, and then ejaculatory duct, and from there the particularly ejaculation can occur. So according to 17th question, option 4 is what correct here, right? Then the actually like what, select the what, mismatched pair here, right? Select the mismatched pair, and that what we have to find out the answer with respect to that. So if you consider first month of what, the pregnancy, that is what, formation of heart, this is actually correct, right? Then the second month of pregnancy, movement of what, fetus, right? So in a second month of pregnancy, actually still the digit development is going to take place, but here the movement of the fetus can be seen with respect to the what, fifth month of what, pregnancy, right? So fifth month of what, the little movement of what, the particularly fetus can be seen. Then the third month of what, pregnancy, formation of most of the major organ systems, that what we are going to see, that is also correct. Sixth month of what, pregnancy, eyelid separates and eyelashes are what formed, so that is what we are going to find out. So overallly you can consider here the incorrect or mismatched is nothing but that is what, B, that is movement of fetus with respect to the what, second month of pregnancy. Majorly it takes place with respect to the fifth month of what, pregnancy. Then 19th question, that is out of the following option, identify which one is not a natural method of contraceptive, which is not actually a natural method of what, contraceptive. So natural method of contraceptive, what you are going to find out, lactational amenorrhea, right? So when the particularly mother delivers a baby, right? So during that period, the female up to the what, on an average 6 months, there will not be any menstrual cycle, right? And as the mother is feeding the baby, so that what we are going to call it as the first natural method of what, contraceptive, that is lactational amenorrhea, right? Then periodic absentia, by looking at with respect to the menstrual cycle, we know is that 14th day of a menstrual cycle we are going to get what, ovulation. So before the particularly what, the 10th day, and after like what, 18th day, right, of what, menstrual cycle, that what actually we call it as periodic absentia, or majorly with respect to the 10 to the 18th, if you won't perform a coitus, that itself what we call it as periodic absentia, by knowing and understanding the cyclic changes with respect to the what, woman, right? Then the particularly coitus interrupts, right? So that what you consider majorly we won't allow the particularly meeting of this particularly sperm with respect to the what egg, that is what we call it as coitus interrupt, removing of what the particularly penis from the what vagina before the particularly sperm get releases, right? Then implants, so majorly this is known to be the what we call it as barrier method, that what we are going to find out, right? So implants, which is actually inserted with respect to the what skin, right? So here one is not actually a natural method of contraceptive, that is nothing but what implants. Remaining all of them are what the natural method of what contraceptive, right? Then in zygote intrafallopian tube transfer, the embryo up to dash stage is transferred into the what fallopian tube. Majorly with an 8 blastomere, that what they are going to transfer, right? Then there will be what overall development, and we can even find out the suitable of that zygote with respect to the what the particularly female uterine cavity, right? So suitability can be what checked over there, right? Then the majorly what 8 celled blastomere what they are going to use. Then the particularly 21st question, that is read the following statement and answer it accordingly. So MTP statement 1 what says is MTP is to get rid of what unwanted pregnancy due to the casual unprotected intercourse or failure of what contraceptive used during the coitus or rapes, right? Then statement 2, right, what is that? MTPs are performed legally by qualified doctor by giving a proper medical justification. So here both the statement 1 and 2 are correct, right? Then how many types of what, 22nd questions, how many types of what gametes will be formed by a particularly parent with genotype? They have mentioned genotype A, capital A, small b, capital B, small b, capital C, small c, right? So majorly to find out the gametes, we are going to use the formula, that is 2 raised to n, right? And majorly what this n stands for what here? Heterozygous condition, right? So how many heterozygous conditions are there here? 1, 2, 3, right? So with respect to that 2 raised to 3, that is nothing but what 8. So here answer is the 8, right? So that those many like what particularly gametes can be seen with respect to this heterozygous of what parent, right? The conditions what they have mentioned, right? Then when a single gene exhibit multiple phenotypic expression, the phenomenon is called as, majorly this phenomenon is called as pleiotropism. The best example for the mainly what pleiotropism is phenylketonuria, right? Based on the absence of only one gene, we can get what majorly so many symptoms or so many types of what characters with what this particularly phenylketonuria. So here answer is what pleiotropism, that is single gene exhibits multiple phenotypic expression, and this phenomenon is called as the particular pleiotropism. As I have told you, example is phenylketonuria based on the absence of what phenyl hydroxylase enzyme, right? Then the particularly person will suffer from what albinism, he'll even sometimes so like what mentally becomes retarded. So this kind of what more phenotypic expressions or characters what we are going to see with respect to the pleiotropism or pleiotropy, right? Then a color blind man marries a carrier woman. The percentage of their color blind progeny in the what next generation will be. So how we are going to find out? What they have mentioned, a color blind man marries a carrier woman. So if you consider with respect to the man, it is X-linked disorder. Then if male itself is affected or male is a color blind, then I'm going to find out what XCY, right? Then with respect to the carrier, female carrier, what I'm going to mention here, the particularly XCX, she is a carrier, right? So if she is a sufferer or she is carrying a disease, means how I'm going to represent XC like this, understanding? Then only she is actually suffering from what color blind here, right? Then overall with respect to these expressions, if you find out, right, so how much percentage I'm going to get here, right? Here majorly I'm going to get what 50%. So majorly with respect to the Punnett square, we can understand now here two types of gametes can be formed, that is XCY, right? Here also what I'm going to get here XCX, two types of what gametes. So 50% will be the carrier and 50% will be the what non-carrier. So if I consider, if I go on crossing XCX, right, this is one, here XCX, this is one more, right, here XCY, understanding, and then here XY, right. So majorly if you consider, this is actually color blind, right, this is color blind, and here also this is color blind, right. But these two, this is a carrier actually, what it is, a carrier, and this is normal, right. So majorly one female is carrier here and one male is what, normal here, but two of them are what, the particularly color blind. So according to this, if I find out, the particularly 50% of them will be the particularly whatever this color blind progeny, and 50% will be the what, normal. Inside that normal, one is just a carrier. So she is not got affected with respect to the what disease, but she is just carrying a that particular type of what genes here. So we'll go ahead with the what next question here, right. So majorly identify which one of the given pair of option 1 is correct with respect to the what Down syndrome and Turner syndrome. So we should actually match up the things, and then only we have to answer such questions. Majorly what I know, Down syndrome, short statured individuals, yes they are, right. But with respect to the Turner syndrome, symptoms what they have mentioned is gynecomastia in man. That actually we are going to see it through whom? The Klinefelter type of what symptoms, right. Then round head, partially open mouth, overall masculine development. This masculine development is with Klinefelter. Then broad palm, physical and mental development, right. Mental development or retarded, right. So with respect to the what sterile females and majorly what rudimentary ovaries. Then additional copy of an X chromosome, that is not going to take place, and absence of what, an X chromosome, that is what we are going to see over here with respect to Turner. But matching what if I find out, that is short statured individual with respect to that what gynecomastia with respect to the what man, that is what the particularly correct match here. Option is what, particularly 3, right. Then RNA polymerase 1 is responsible for the transcription of RNA polymerase 2 is responsible for the transcription of what, mainly hnRNA, right. This RNA polymerase 1, 2, 3, right. RNA polymerase 1, 2 and 3, right, which actually shows what clear cut division of what labor with respect to the only eukaryotic. So only it can form what, hnRNA. hnRNA cannot be produced inside prokaryotic, right. So here hnRNA can be found out with respect to the word only eukaryotic. So here RNA polymerase, and what you should remember, in prokaryotic only one type of RNA polymerase you are going to get, not these particularly 3, 1, 2 and 3, right. So here hnRNA can be produced by whom, the particularly here the RNA polymerase 2. So with respect to this, answer is what, 1, right. But snRNA if you consider, snRNA as well as the particularly tRNA can be produced by whom, the particularly RNA polymerase 3, and the particularly rRNA is produced by whom, the particularly RNA polymerase 1, right. So like that you are going to find out, right. Then so next 27th question, that is which of the following enzyme increases the permeability of the particularly bacterial cells in a lactose. So this question is related to what, the lac operon concept, right. It is related with what, lac operon concept, and with the structural gene what we know is Z, Y and A is going to produce what, Z is going to produce beta galactosidase, right, and Y is going to produce something called as what, permease here, understanding. So gene expression study, and the A is going to produce something called as trans-acetylase. It is going to produce trans-acetylase. So if you find out with respect to the what, the particularly what they are asking, enzyme increases the permeability of what, bacterial cell to lactose. So lactose should what, it should get enter, right? Lots of what more amount of what lactose should get enter, so that it can carry out what lactose metabolism. So who is actually going to carry out lactose metabolism here? The particularly beta galactosidase, right? Who is going to carry out beta galactosidase? So lactose will get converted into glucose and the particularly galactose here, glucose and galactose. So that what who are going to do, that is beta galactosidase. But who will give the permissions? That is what the permease name itself will tell you. So permeability, permease enzyme, right? So that is what 27th answer is the permease, right? Then you consider 28th question, right, which is again related with respect to the what the mRNA and even how the actually protein synthesis can take place. So which of the following statements are correct with reference to the what prokaryotic genome? So here the prokaryotic genome, the particularly structural gene is known to be monocystonic structural gene. No, it is not known as monocystonic, because with respect to the single mRNA, there will be attachment of what many ribosomes on it. So this condition we mainly call it as what polyribosomes. And diagram when we draw with respect to the bacteria, this we are definitely going to draw. That is nothing but polyribosome condition that can be seen with respect to the word prokaryotic, right? So with the single mRNA, it can produce many proteins. So definitely I will call it as polycystonic, not the particularly monocystonic here, right? So here that is monocystonic structural gene is actually not correct with respect to the what? The particularly prokaryotic genome. Then introns absent in structural genes, right? Introns absent in what the particular structural gene. So this statement is correct. So there will not be any introns because there is no any splicing or formation of a HN mRNA or HN RNA is not going to take place with whom? The prokaryotic, right? So here intron is absent in structural gene. So this is actually correct here. Then transcription and translations are coupled process here. Yes, these are what coupled process, right? One way there is a formation of mRNA and then next there will be attachment of what this particular ribosomes you are going to find out which can carry out translations. It is actually coupled process. Then primary transcript undergoes splicing. No, this is going to take place with respect to the post transcription modification in eukaryotes, not with respect to the what prokaryotic, right? Only one RNA polymerase is present. Yes, only one type of what RNA polymerase, that what you are going to find out with respect to the 26th question, that what I have told you, right? So you can compare this majorly here B and the particularly C and E is correct here. So option 4 is correct, that is B, C and E are correct, right? Then the next question that is related with respect to the what, when change in the gene frequency of a population occurs by chance, suddenly if it occurs we call it as what, the genetic drift. By chance if it occurs we majorly call it as here genetic drift, right? So next question, that is majorly what, the 30th question, right? So Darwin's pinches represents one of the best example by seeing the particularly variations of the particularly pinches, right? One is blunt, one is pointed, so different, and that what it shows? It shows majorly what, adaptive radiations. What it is going to show us? The adaptive radiations. Due to the adaptive character only, the change in the what structure what the particularly organism has brought, that is what the Darwin's pinches represents one of the best example of what, adaptive radiation. A direct question it is. Then choose the word correct statement from the following. So Charles Darwin traveled around the world in a ship called as what, HMS Beagle. Yes, this is what actually correct with respect to the Charles Darwin, right? Then there has been a gradual evolution of what, life forms. Yes, this is also what actually correct, right? Then according to the Darwin, fitness refers to the what, physical fitness only. No, right? So everything matters over there because survival of the fittest matters, right? Then the fossils are remains of what, hard part of what, life forms found in rocks. So this is not even what the correct over here, right? So sorry, this is correct actually, understanding? So Here the particularly fossils remains of what a hard part of the what life forms found in the rocks. That is what correct here. Then Hugo de Veris, a naturalist, worked in Malaya archipelago, right? So here this is also what wrong. So here answer is nothing but what A, B and D are what correct, right? So that is what you are going to find out. In 32nd question, in which of the following HIV replicate and produces its progeny, right? Progeny viruses. Mainly HIV virus goes and affected to whom? The particularly macrophage cell as well as what this particularly T helper cell, or mainly the T lymphocytes. Mainly even we can represent it as what? CD4 cells, right? So CD4 cells of what? T lymphocyte, mainly called as what? T helper cells. There the HIV get replicates and produces its progeny viruses, right? Then which of the following are the techniques for detecting the particularly cancer of what? Internal organ? So mainly here if you consider the matching Widal test, they have mentioned here also, they have mentioned Widal test. So Widal test is used for what? The particularly typhoid, right? So here remaining whatever these two options are, what wrong? Then what left out? The particularly A, radiography and MRI, and MRI and computed tomography. With respect to that, A and B is what actually corrected technique for the what detection of what cancer of what internal organs. Then malignant malaria is caused by whom? The particularly majorly who? The Plasmodium falciparum, which is known to be the what dangerous parasite which causes what this particularly malaria, majorly protozoa Plasmodium falciparum, right? Then the drug prescribed to the what patient who undergone organ transplant and dash and is produced by, right? So majorly for the organ transplantation, with respect to the what the particularly patient who went on the organ transplantation, that particular person have to take a continuous immunosuppressant tablets for what whole life. So here immunosuppressant tablet is what? Cyclosporine, right? And it is actually produced by whom? The particularly Trichoderma polysporum, right? So here answer is what? The particularly 1, right? So next question, that is 36th question, right? So read the following statement and select the correct options. So statement 1, what it says, biocontrol refer to the use of what? Biological method for controlling plant disease and pest. Yes, biocontrol actually is a technique used for what? The particularly controlling this particular disease and the pest. So there what they are going to use, this particularly Trichoderma species are effective biocontrol agent for several plant pathogens. So that Trichoderma, the fungi which can be used as particularly biocontrol agent here. So here both the statement 1 and 2 are correct, right? Then match the column 1 with the column 2 and choose the what correct option given below. So they mentioned what here, the particularly different organism like Streptococcus, Penicillium, Methanogens and Anabena. So which you will find out what easy, first you match that, right? And if you are not getting what answer with respect to the word Streptococcus, but you know is like what Methanogen and Anabena. Methanogen is known to be the word biogas production, right? And Anabena is what? Free living nitrogen fixing. So I have to see the options now. How I am going to find out? Consider D is 1, right? So here I consider D is 1, here also 1 is there and here also 1 is there. Then I have to see for what? Methanogens, that is what? Biogas production. Then C with respect to the methanogens, what I am finding out? The here also 4, but here it is 3. Understanding? So here the correct answer you can mention it as what? The particularly 3, right? Because the penicillium is a what? Source of antibiotic, and the Streptococcus which produces what? Stannine, known to be what? The statin, the clot buster, right? So like that what the matching can be even performed. So 38th question, that is match the content with list 1 and 2, right? So here they have mentioned what? Bioreactor, for what it can be used as? Here the vessels which convert the raw material into a specific product. It's a large vessel, right? So that's what we call it as bioreactor. Downstream processing, for what it is? A purification method it is. So that is what here, which involves the separation and the particularly purification. Then recombinant protein, how we can produce a recombinant protein? The insulin produced by what? The particularly rDNA technology, right? Then PCR is to what? The majorly detect the mutated gene in a suspected cancer patient. So here the answer is what? 3 here, right, with respect to the word matching this particularly following. Then the part of a plasmid that code for protein and involved in the replication of what? PBR322 plasmid is, right. Majorly you consider cloning site where I am going to add particularly gene of interest. Then ORI, which actually help in something called as what? Replication, but it won't code for any kind of what? Protein. Then selectable markers, majorly we call it as antibiotic resistance genes, right. So here the answer is what? Rope, right, rope, which actually code for protein and even involve inside what? The replication of what? This particularly PBR322. PBR322 means the plasmid of Bolivar and Rodriguez. So next question here, that is the particularly, let me check with the previous one. Yes, right. So next, that is the 48th question. To isolate DNA from fungal cells, bacterial cells and plant cells, the enzyme required are respectively, right. So we have studied related with biotechnology principles. There you have came across fungal cell is made up of what? The particularly actually chitin. It is made up of what? The chitin. And bacterial cell is made up of what? Peptidoglycan, right. Then the particularly here plant cell is made up of cellulose. So based on the word, the name itself, you can imagine that is chitin is for chitinase enzyme. So it can be degraded by whom? The particularly chitinase. Then the peptidoglycan can be digested by what? Lysosomes or lysozyme, not lysosomes, lysozyme. Then the cellulase can be digested, cellulase can digest what? Cellulose here, right. So like that you are going to find out. So here option is the particularly first one is correct, right. So this is the first one, option is correct over with 40th question. Then the particularly majorly 41st question, that is in mature insulin which of the peptide is not present, that which peptide is not present, that what you have to find out. In the immature insulin, what I am going to find out, it is made up of actually what? 3 chains, right. One is like what? B chain, then A chain, in between of that what? C chain. This I am going to call it as immature type of what? Insulin, right. Immature. But the matured form of what? Insulin, during that period what is going to happen? This C peptide is actually removed and A and B are going to form a disulfide bridge between them, right. So joining of A and B, so that is what? Mature insulin. So what they are asking with respect to the mature insulin, which peptide is not present? So which peptide is not present? C peptide, which is actually what? Removed. So that is what here answer is C peptide. Understanding? Then a scientist wants to produce virus free plant in a tissue culture. Which part of the plant will he use as an explant, right. Majorly to select, when you are selecting explant, you should find out the part which is actually in continuous division. So it should be what? Continuous in division, right. So it should have what? Group of young cells, right. So which are always in divided. It should have what? Group of young cells. So definitely this particularly scientist will select that only. So they have given what? Mature stem. Here there is no any group of what? Young cells, as it is become what? Mature itself, right. Then axillary meristem, yes, this can be selected because there the cells are actually in continuous division, axillary bud related, right. So which will sprout up and form what? Something called as new part of the plant. Then apical meristem, which is actually present at the tip of root and tip of the shoot. So there also what we'll find out? Continuous dividing cells. So the next mesophyll cells. Now well developed leaf itself has what? The particularly mesophyll cell. So here it's not what? Mature stem cells, or even it is with respect to the what? The particularly mature stem, or with respect to the mesophyll cells. So here answer is what? Axillary meristem and apical meristem, what he is going to choose here. So here that is the particularly B and C is correct. Here what it is? B and C, the fourth one is correct. Then some strains of what? Bacillus thuringiensis produces protein that kills insect. So which one of the following is not killed by the protein of Bacillus thuringiensis? So majorly what we know, this Bt toxin, a protein, right, which is going to kill what? Lepidopteran, cleopteran and diopteran type of what? Insects. The mosquitoes, insects or the flies, that what actually whose protein, this Bacillus thuringiensis protein can kill. So here if you consider cotton bollworm, tobacco budworm and armyworm are the example of what? Lepidopterans, right? So tapeworm, which is related with respect to what? Majorly human, right? Or even with respect to the animals, what we are going to find out. So here this cannot be killed by means of what? Bacillus thuringiensis, right? So which one of the following population attribute contribute to increase in the population density? So how actually population density can get increased? One is by means of natality, that is what? Birth rate, and even immigration, right? The people which they were coming from what other to the what? This particularly one location, other location to the one location. There the birth rate and birth rate or majorly can be called as natality and immigration can affect the particularly increase in the population density, right? Then they mentioned what? If 8 individuals in a laboratory population of what? 80 fruit flies, right? So here they mentioned it as what? Total fruit flies are what? The total fruit flies 80, right? First moment, right? And they mentioned what? 8 individuals. Now we have to see what has happened with respect to this particularly 8 individuals, right? So that what we are going to find out. So if you consider, what they are saying is 8 individuals in laboratory population of what? 80 fruit flies died during a specified time interval, and the death rate in the population during that period is, right? So here what these 8 individuals, these actually died, understanding, and what we have to find out? The particularly death rate. So to find out death rate, what I'm going to use here majorly, like how many individuals died with respect to that particular total number. So 8 divided by 80. So majorly what answer I am going to get is 0.1, right? So here 0.1 individual per time interval, that will be the what actually answer here, right? A simple question, right? Then next, that is 46th question. So choose the word correct sequence of the word step involved in decomposition. So what is going to happen first with respect to decomposition? The large components should get what? Decompose. So first decomposition we call it as fragmentation, right? Following to that, what should occur? The leaching, some components to be what? Added in. Then there will be a catabolism, then humidification, and then the actually mineralization can take place. So your answer is what? 2. Then 47th, right? So with respect to the what? Limitation of what? Ecological pyramid, which of the following statements are correct, right? So ecological pyramid basis, what we have to find out? Limitations of what? Ecological pyramid. It does not take into the account of the what? Same species belonging to or more tropical levels. Yes, this is what actually correct. Then it assumes a simple food chain, something that almost never existed in nature. Yes, this is even correct, right? Then you consider it accumulate the saprophyte. No, it won't accumulate saprophyte. Then it does not accumulate the food web. No, it does not accumulate. So this is what actually correct. So you can consider which is a limitation of what? Ecological pyramid. With respect to that, A, B and D is correct. Understand? Then the 6th extension of species represents in the progress is dash time faster than the previous 5 episodes of what? Mass extension. So they talked regarding what? The particularly 6th extension. So here it is, answer is what? 100 to 1000, right? Times faster than the what? Previous 5th episodes of what? Mass extension, right? Then species diversity dash has move away from the what? Dash towards what? The species diversity decreases as we move away from the what? The particular equator towards what? The actually poles. If we start moving towards what? Consider this is Earth. If we start moving towards the particularly pole, the particularly what is going to happen? Decreases with what? Equator when we reach towards what? The particularly pole, right? So population will actually decreases. Understand? So here, that is the 49th question is over. The answer is what? Decreases equator and even poles. That is 4th one. So 50th one question, that is, in practical examination, the following pedigree chart was given as a spotter for the word identification. The student identified the given pedigree chart as, right? So by seeing this, what we feel is it is actually recessive in nature, because it is skipped one generation here, here also it's skipped, right? So we call it as what it is actually the recessive one, right? But what I am finding out here, both of the individuals are affected, understanding? So here one way, here the particularly character, this both of the individual is not what they have not carried the particularly character, but it passed on to the what male, right? Such kind of what you can find out, mainly we call it as autosomal recessive one. It can be called as what? Autosomal recessive one. It is not related with X-linked recessive. Why? Because you consider X-linked, if it was there, then the male should pass on, the female should pass on to the what male, and some way in between the female should be what carrier with respect to the what specific type of what generations, and may even can skip the particularly generation, right? So your answer is autosomal recessive. A student observed the particular TS of a plant organ slide under the microscope. He observed vascular bundles in the particularly stellar region as conjoined, collateral and open, right? So with respect to the experimentation, what they are talking about. So what they have observed, vascular bundles in stellar regions as stellar means where the arrangement of what vascular bundles with respect to the what the endodermis, right? So there what we are going to find out stellar region as conjoined, collateral and open type. Based on these features of vascular bundle, identify the correct option below. So if there is a presence of vascular bundle in stellar region and they are conjoint and collateral, conjoint, collateral and open type, then definitely it is what? The dicot stem, right? Then a student observed the slide of what mitosis under the microscope and observed that the chromosomes were placed at the opposite poles, and which stage, if he observed, like maybe he observed like this, the chromosomes which are what to the opposite poles, right? So like this, the structure. So with respect to this, if you find out this stage, I can identify it as what? Anaphase. This stage you can identify it as what? Anaphase, right? Then 53rd question, identify the correct statement with respect to the word rules of what binomial nomenclature. So we should find out the word incorrect statement with respect to the word binomial nomenclature. So biological names are underlined separately when handwritten. Yes, this is actually a correct. Then biological names are printed in italics to indicate their non-Latin origin. No, they are of what? Names are what? Latin origin. They are mentioning it as what? A non-Latin origin. So this statement is incorrect, right? Next, the first word represents the genus while the second word denotes the word specific epithet. Yes, this is correct. Then biological names are generally Latin or Latinized irrespective of what their origin. So here the particularly incorrect statement is what with respect to the binomial nomenclature, that is second one. Biological names are printed in italics. Yes, they are printed in italics to indicate their Latin origin, not the particularly non-Latin origin. So statement 2 is actually incorrect, related to whom? The particularly binomial nomenclature. Then match the column with the column 2 and choose the word correct option. So they have mentioned bacteria and you have to find out the word particularly shape, and based on the shape only these names are, right? So coccus means majorly what it is. Coccus is nothing but what it is, spherical, right? Then bacillus is nothing but what, here it is spherical. Then bacillus is rod shaped, vibrium is what, comma shaped, then spirillum is what, spiral shaped. So according to this, if you find out the particularly fourth option is what, correct, right? Then read the word given statement and choose the word correct options here, right? What they have said is statement 1, the gamma are green unicellular sexual bud which develops into a receptacle called as gamma cups. No, they are gamma is what? These are the unicellular asexual buds. Majorly these are what? Asexual buds which develop in what? Receptacles called as what? Gamma cups. So this statement is wrong, right? Then statement 2, protonema develops directly from the spores. Yes, that is what actually correct. Protonema, spores to the word protonema which forms a filamentous structure, then it will form what? A leafy stage. So here the statement 1 is false, but the statement 2 is true, right? So during the field trip, a student observed a marine organism with a worm like body. Right. The cylindrical body was divisible into the what proboscis, collar, and the long trunk, and the organism may be known to be balanoglossus. Right. Balanoglossus. So based on this particularly, it is answer is what balanoglossus. So we have to find out what 57th question, identify the particularly estivation type, right? So if you find out it's a axillary type, it is imbricate type, and it is twisted, and it is what velvet. So with respect to that, answer is what here, the one, right? Then match the column with respect to the word column 2 here. So whatever this particular structure what they have mentioned here, if you consider radical tetrarch cambium ring between xylem and phloem, that what I'm going to find out with respect to the TS of dicot root. Then conjoined open end arc type, that I'm going to find out with respect to the TS of dicot. Radical polyarc large with pim cambium ring, then that what I'm going to find out with respect to the monocot. And the particular conjoint, that I'm going to find out with TS of what the particular monocot. So here answer 1 is correct, right? Then which of the following statement are correct with respect to the frog, right? So here majorly frog has what? The Beeder's canals are present in the male frog. Then even sound producing vocal sacs are present in the particularly male frog. So here option 1 is correct, right? Then the reserve food material in prokaryotic cells are stored in the cytoplasm in the form of what? In the form of majorly inclusive bodies, right? So this is all about the particularly KCET 2025 question paper. You compare the answers and find out the key answers and check how much you have got, right? So all the best for your future event, right? Thank you so much, and please do subscribe our channel and